Let's begin looking at the origin of cholecystokinin, or CCK. CCK is going to be released upon sensing digestion products because the purpose of CCK is to increase the secretions of pancreatic enzymes. And so the presence of dietary components is going to be the trigger source for this. So we have to look at two particular cell types that lie in the intestinal epithelia. And this, of course, is occurring early, so it's in the duodenum. And the first cell type is going to be a cell type that is going to sense the presence of amino acids. Now, of course, this is the lumen up here, and so these amino acids are coming from dietary sources. The dietary source is protein. The protein is being digested by proteases. The first protease involved is going to be pepsin in the stomach, which of course is activated from pepsinogen by low pH. And the second one is trypsin. Trypsin is one of the major proteases put out by the exocrine pancreas. Now, what triggers this trypsin release? Recall from our regulation of pancreatic secretions, this trypsin is coming from originally the cephalic phase of activation. Because the intestinal phase, where we're going to get the bulk of secretion of digestive enzymes from the exocrine pancreas, is going to be due to cholecystokinin stimulation, CCK. This process here, we are getting the cholecystokinin into the bloodstream. So this trypsin is that little bit that comes out in the cephalic phase. So once we have these amino acids, they bind to receptors, they send signals through here, and they are going to release a small protein called CCK releasing peptide. Now this small peptide is going to bind to another cell type, again on the intestinal epithelial layer. These are eye cells, or intestinal cells. And the CCK releasing peptide will bind to its specific receptor here. Now this is not sufficient enough to induce the secretion of the hormone cholecystokinin. We need another factor. And this other factor is coming out of the exocrine pancreas due to the cephalic phase stimulation, and this is called monitor peptide. This binds to its receptor, and now once we have this dual signal between the CCK releasing peptide and monitor peptide from the pancreas, now we can secrete CCK into the blood. Once the CCK gets into the blood, of course, it can travel to the pancreas and induce the intestinal phase of secretion, which is fusion of the vesicles inside the acinar cells that release all of the digestive enzymes. The other thing that CCK is going to do is to relax the sphincter of Oddi. So CCK binds to the cells of the sphincter of Oddi, the smooth muscle cells, and induces a relaxation. Makes sense? In order for the pancreatic secretions to be able to be put into the duodenum uninhibited. The other thing the CCK does is it causes contraction of the gallbladder. When we talk about the liver and the gallbladder, we'll see that the gallbladder is the storage organ for bile, and bile is going to be used for the emulsification of fats. Again, CCK essentially is the major hormone to induce the intestinal phase. And as Dr. Shuttleworth mentioned, CCK also reduces stomach emptying. And this is to control the rate of nutrient delivery into the intestine. So we slow down the stomach emptying and allow what's in the intestine to be processed for most efficiency. Now, the regulation of this process is self-regulating. Recall that we have trypsin here. The trypsin can digest both CCK releasing peptide and monitor peptide. When trypsin degrades these, the signal goes away. So what prevents the trypsin from prematurely degrading this before it has an effect to get the CCK out? Well, remember that we have lots of dietary protein. The dietary protein is a substrate for trypsin. CCK-RP and monitor peptide are also substrates for trypsin. When we have a lot of dietary protein, that will consume the trypsin's activity. And it will outcompete these peptides from being degraded. So with lots of dietary protein, we have protection against trypsin degrading our signal that is secreting CCK. As our dietary protein reduces because of digestion has come to a close, come to an end, now the trypsin has access to these, digests them, and it reduces and eliminates the signal for downregulation. The other hormone we have to look at is secretin. Now recall, secretin, major purpose of secretin is to increase bicarb coming from the exocrine pancreas, plus increase the volume of fluid. Increasing the volume of fluid is adding water to our digestive material, and it's also washing out the pancreatic ducts. So what triggers the release of secretin? If we look at a graph where we're comparing pH, with the amount of bicarbonate ion from the pancreas, what we see is that at high pHs, we have very little bicarb. As our pH starts to decrease, meaning more acid in the solution, we see that the rate of bicarb appearance in the duodenum begins to increase. So this is suggesting that the mechanism of secretin release is due to a pH-sensitive receptor. And this is the case. The cell type is an S cell, standing for secretin. And the S cell will recognize and detect, essentially quantify, the concentration of acid. When the acid concentration is high proton concentration, the S cells will now stimulate the secretion of secretin. And then, of course, the secretin will increase the release of bicarb and water into the intestine.